Thank you very much, Mr. Chairman. It's a pleasure to be here. It's a pleasure to debate against an eminent person like Warren, and I'm sure that he convinced you that his legend is true. But let us look at it a little bit more seriously. Actually, you know that this is an old story, led by Professor Prusiner many years ago. This is an 87 paper, 10 years before he was awarded the Nobel Prize. He did not get the Nobel Prize for this, only for prion diseases. But he continued and to develop the concept and convinced Warren and many others that it's not just prion diseases, and maybe he should be awarded the second Nobel Prize. So we'll see. So is this, now we are facing this issue of prionization of all neurodegenerative diseases. So it's even genetic diseases like Huntington disease and many other diseases that are being declared to be prion diseases. And the question that we have to answer is whether it's a fact or is it just a myth. And I would convince you, I'm sure, that it's a legend. We're all clinicians, or most of us, so let's compare firstly clinically what we all know, the big differences that exist between Parkinson's disease and Creutzfeldt-Jakob disease, the most important, most common prion disease. Firstly, in terms of evolution, very rapid in terms of Creutzfeldt-Jakob disease. People die within a few months, whereas in Parkinson's disease, we speak of decades of the development of the disease, decades that it takes the presumed alpha-synuclein to go around and to infect other cells. Why does it take so long? Of course, in Creutzfeldt-Jakob disease, it is not a single system which is affected, but rather many systems. So the phenotype is heterogeneous rather than in Creutzfeldt-Jakob, rather than similar in Parkinson's disease. EEG changes are dissimilar, and the pathology is quite unlike one that we see in Parkinson's disease. So in Creutzfeldt-Jakob, we see the spongiosas, which we don't see in Parkinson's disease. We see astrogliosis, which does not exist. And very importantly, the cells which are affected are non-specific in Creutzfeldt-Jakob disease and the other prion diseases, but are specific cells that are affected in Parkinson's disease. So what are the elements? Let's look at it seriously, step by step, and ask ourselves, what is going on? So the elements of the prion theory as applied to Parkinson's disease are first, that there is an initial stochastic change of native alpha-synuclein to what we can call alpha-synuclein star. And then that alpha-synuclein star serves as a template for further conversion of native alpha-synuclein to more alpha-synuclein star. That this alpha-synuclein star is carried along neural pathways. And finally, that alpha-synuclein is toxic to cells. So let's look at them one by one and see whether these are correct, whether this can be verified. So the first one is that there is an initial stochastic event that changes the native alpha-synuclein to alpha-synuclein star in parallel to prions that are changed from the PRP-C, as we call them, to PRP-scrapy, as we call them. So we've not heard about that at all. What is it? And of course, this stochastic change occurs in Creutzfeldt-Jakob disease, can occur all over the place. Can take place in the cortex, on the right or on the left, in the front or in the back, can take place subcortically in the basal ganglia or in the thalamus, can take place in the cerebellum. This is what we expect from a stochastic event. The suggestion in Parkinson's disease, it occurs in one place, only in one place, only in the dorsal motor nucleus of the vagus. Why just there? Why would this stochastic event, so I think what we heard from Warren is that maybe it's not a stochastic event at all. Maybe there is a some toxic material that causes this thing to happen. Let's hear what this toxic material is. We don't know what it is that affects the olfactory bulb and the dorsal nucleus of the vagus. But anyway, it is not a stochastic event. So it's unlike prion diseases. So in prion diseases, the initial change may occur randomly in neurons, whereas this is not the case in Parkinson's disease. Now, who is the villain? Who is this PRP scrapey equivalent? So we hear something about misfolded alpha-synuclein. So is it misfolded normal alpha-synuclein, or is it something which is the native one, or is it should be phosphorylated or acetylated? Are these monomers or aggregates or fibrils or ribbons? Who are these villains that go along this neural pathway to infect other cells? Let's find out who are they, and then we can perhaps look at them and try to eliminate them. We never heard what these are. We've never been shown which of these is the important one. So let's move to the second point, because I hope I convince you that the initial stochastic change of native alpha-synuclein to alpha-synuclein star is not what happens in Parkinson's disease. But the next basis of the prionization theory is that alpha-synuclein serves as a template for further conversion of native alpha-synuclein, and you have already seen this slide. But I want to point out, what did you do with this pointer? Did you take it with you? Come on. This is a little bit unfair, but okay. So in prion diseases, this can be shown very easily. It can be shown in vitro. It has never been shown for alpha-synuclein. Never, ever. You cannot show in vitro that alpha-synuclein, once transformed into the alpha-synuclein star, okay, will go on to promote this further. This has never been demonstrated. So it's not true. It's not true that alpha-synuclein has been shown, at least, to serve as a template for further conversion of native alpha-synuclein. So the third basis is that alpha-synuclein star is carried along neural pathways. Which neural pathways? So the first deposition of alpha-synuclein is in the olfactory bulb, according to Brack. Where does this go to? What pathway? What are the next steps in this presumed hypothesis? Nothing. There is no next step from here. There is a next step from the dorsal motor nucleus of the vagus to the dorsal raphe and to the cerulus. Now, I was very good in anatomy lessons. I never heard about this pathway. There is no such pathway leading from the dorsal motor nucleus of the vagus to the dorsal raphe or to the locus cerulus. However, there is a pathway that goes from the midbrain, from the substantia nigra, into the basal ganglia. But the basal ganglia are not affected in Parkinson's disease by alpha-synuclein deposition. There is no alpha-synuclein deposits in the striatum, even in advanced cases of Parkinson's disease. So this means that the transmission of the is assumed to be carried along by a non-existent pathway, such as that leading from the dorsal motor nucleus of the vagus to the dorsal raphe, but not along existent ones like the nigrostriatal tract. So, in prion diseases, cell-to-cell transmission does not take place primarily through synaptic transmission at all, but rather it spreads to neighboring cells, and this leads to the formation of the status spongiosum, that we'll show later on. So alpha-synuclein star is supposed to be toxic to cells. This is what we are told. But nothing happens like we see in the in prion disease, where you see the status spongiosus. This does not happen in prion disease, in Parkinson's disease. Neighboring cells are not affected. Distant cells are affected. So the spongiosus is the result of PRP scrapie toxicity to neighboring cells, but in PD there are no lacunes, and that is no damage to neighboring cells. Thus, on each of these critical elements, Parkinson's disease is different from prion diseases, from the classical prion diseases. And so what about these impressive lab results that we have heard about? I mean, most of the talk that we heard before from Professor Olanov show that what's happened in animals, not in humans. Not everybody agrees, and we should remember that there are many papers who supported it, but not all of them. This is a paper, an important paper by the work of this gentleman, Stanley Prusiner, and he talked about propagation of prions causing synucleinopathies in cultured cells. This is, as we heard from Warren, mainly taken from MSA cases, but they actually show in this paper that attempts to demonstrate alpha-synuclein prions in brain homogeneous from PD patients were unsuccessful, and not for the lack of trying. So what happens? What do we do with all these lab results? So I think that these interesting lab findings are really an artifact in many ways. They demonstrate alpha-synuclein spread, but this may represent an artifact caused by exposure to excessive doses, excessive concentrations of alpha-synuclein star or alpha-synuclein, exceeding the cell capacity to eliminate them. Because let us not forget that misfolding is a normal phenomenon that occurs in each cell of our body all the time. Proteins are recycled all the time and are misfolded all the time. And the organism over the evolution has developed methods to deal with them. And Dieter Meyer just recently told us that this is the autophagy mechanism. Now, of course, this mechanism has a capacity, but not an unlimited capacity. And if overload it with whatever, then there will be consequences. These are toxic consequences. These are not pathogenic consequences. This is a different story. So all these experiments that Warren Olno went along to tell us were all done in these artifactual, not natural circumstances. They're all used either animals having overexpression of alpha-synuclein or exposing them to huge amount of alpha-synuclein which were injected to them. Also, in most of these experiments that Warren has shown you, there is no cell death and no relevant clinical phenotype. So, you know, they do not develop Parkinsonism, and we know that animals can develop Parkinsonism. So, and there is still missing, Warren showed us maybe one example, but this has not been replicated in any other laboratory, that this transmission can be carried on and on and on from animal to animal. So this is an important part of the prion theory, but it's not there. And let's not forget the history. So this is not the first time that this idea came about. Carlton Gajduchek, who got the Nobel Prize for prion diseases, although he did not call them this way, he called them unconventional viruses, but he tried to see whether what he showed in Kuru and in Creutzfeldt-Jakob and in Gerstmann-Strauss-Schenker occurs also for other diseases, neurodegenerative diseases of the nervous system. This is something that is obvious to look for. And unfortunately for him or for us, the answer, or rather for Warren Olanov, the answer is no. So there are lots of studies, and these were done not on rodents. These were done on higher animals. So monkeys and apes could not be shown to develop a disease when injected with brain substance taken from patients dying of Parkinson's disease or of Alzheimer's disease or of ALS. But quite easily if brain samples were taken from patients who died of Korczyn disease or others. Then there is another interesting story that has been shown, and you all know that Korczyn disease can develop spontaneously, can develop genetically, and also iatrogenically, and this does not occur anymore now, but 30 years ago, people were injected, kids were injected with growth hormone, which was derived from pituitaries, which were removed from people who died of whatever cause. And unfortunately, in this large batch of pituitaries that existed there must have been one person who died of Prion disease, and this caused an infection. There are several dozens young children who developed Korczyn disease later on and died of the disease. Now, you would expect that in this mixture of pituitaries, there would also be samples taken from people who died with Parkinson's disease or with Alzheimer's disease. None of the kids, none of the surviving people who did not develop Korczyn disease, because not all of them did, ever developed one of these diseases, as shown in this paper. So in this unique in vivo model of human-to-human transmission, we found no evidence to support concerns that neurodegenerative diseases underlying AD and Parkinson's disease transmit disease in humans, despite evidence of their cell-to-cell transmission in model system, which, as I said, are artifactual. And what about this example that Warren went to some length to tell us? So he showed us that in these transplanted tissues, embryonic transplanted midbrain tissues of the pulmonologic cells implanted into the striatum, after 10 or 20 years, some of the cells develop synuclein deposits. Not all of them, but nevertheless, I mean, how did it get there? Remember that these are denervated cells. There is no innervation. They're not innervated. So the implication that Warren tried to convey, to say that this came from the nigra, how? So, as I said, this is an unlikely explanation. So in conclusion, there are several critical differences between Parkinson's disease and Prion diseases. The Prion theory attempts to explain the spread of the disease, not its origin, whereas in Korczyn disease, it includes the origin. The stepwise progression of Parkinson's disease can have many explanations, but we don't have time to go into this. There is a basic difference between transmissible and transmitted disease. This is critical to understand. The fact that you can artifactually do it does not mean that that's the way it happens in the normal situation. So the fact that you can, even if we accept all these things, all these laboratory results, this does not mean that the sporadic form of Parkinson's disease develops in the same way. And therefore, I conclude that Parkinson's disease is not a transmitted disease, that the Prion theory of Parkinson's disease is full of holes, deserving to be called status spongiosus. And while the results of animal experiments are interesting, they are not relevant to the clinical question that you have to vote on. Is Parkinson's disease an infectious disease? Parkinson's disease is not effective. Thank you for your observation.